Welcome to the continuation of our nervous system. In the last couple lessons, we've been really hitting on the anatomy of the nervous system, specifically neuroglia, which are your support cells, and then neurons. In the next couple lessons, we're going to be hitting on the physiology aspect. So, how does a neuron transmit a nerve impulse to fulfill its function? So, we're going to start by talking about the two most important functional properties of neurons, and that is irritability and conductivity. Irritability is defined as the ability to respond to a stimulus and convert it into a nerve impulse. So remember, a stimulus can be anything such as the room is bright, to the temperature, pressure, anything that you're taking in with your ears, eyes, taste, anything like that, your senses. Conductivity is once you have this nerve impulse, the ability to transmit that impulse to other neurons, to muscles, or even to glands to be able to do the action that you want to do. So one thing about the nervous system is that the transmission of a nerve impulse is said to be electrochemical because there are electrical aspects and chemical aspects to sending an impulse. If we were going to transmit an impulse down a neuron, so that means when I say down a neuron, that's going to be along the axon, this is going to be electrical. If we're talking about transmission from one neuron to the next, this is going to be chemical. We're going to hit on the chemical aspect first because we hit on it a little bit in the muscle system, and then in the next lesson, we're going to hit on the electrical part. So transmission of a signal at a synapse, what happens are chemical neurotransmitters cross a synapse or that gap to transmit a signal from, remember, one neuron to the next. So let's go back over a couple anatomy. Here I have two neurons. I have a neuron here and I have a neuron over here. Hopefully you guys remember here, this is going to be the cell body. These are going to be your dendrites. Dendrites are going to take in a signal. Cell body's going to decide what to do. It's going to generate a nerve impulse and send it down. The purple thing is going to be your axon to the end where eventually it hits on an axon terminal. From that axon terminal, we have to somehow get that impulse from this axon to the other neuron. So that happens right down here at that synapse or synaptic cleft. And this is the area we're going to be talking about today during our lesson. So keep this little image in your head when we go ahead and do it. So we're going to go by this step by step. Step 1. When the action potential, or otherwise known as the nerve impulse, from a previous neuron reaches the axon terminal, this electrical charge opens calcium channels and therefore causes calcium to enter the axon. So what does that mean? Well, when this nerve impulse reaches all the way down here, that means calcium rushes in. So again, here is this image that your book gives you. It comes in, calcium enters. What happens is that calcium causes those tiny vesicles which contain neurotransmitters, that is the chemical aspect of sending a signal, to fuse with the axon's membrane. So here the vesicles fuse with the membrane. So vesicles fuse with plasma membrane. The neurotransmitter is released into that little synaptic cleft, or remember this is called the synapse, which is your little gap. So I'm going to say gap between neurons. So again, neurotransmitters are released. They start to flow across this gap. When they flow across this gap, there's little receptors on the other neuron that these neurotransmitters are going to bind to. So here you see a neurotransmitter bonded to a receptor. So those neurotransmitters go across that synaptic cleft and bind to that receptor that I just showed you on the previous slide. And what happens is these receptor channels open whenever a neurotransmitter binds to them, and this causes sodium to rush into the neuron. So here we have a neurotransmitter bonded. Notice that it is open. So this is an open channel. Here we have a neurotransmitter and it is open. Here there's none bonded, so it's closed, and this one there's none bonded, so that it's closed. Here is an open receptor because it has a neurotransmitter bonded to, and notice that it says sodium is rushing into the neuron. Now, if enough neurotransmitter is released, or in other words, binded, a graded potential is going to be generated, which is just basically a small impulse in a localized area. And if enough gets generated, you have enough stored up energy, an action potential or a nerve impulse will occur along that neuron, and it's going to be sent. Now, these electrical changes prompted from the neurotransmitter binding are very brief. Impulses are sent continuously, rapidly, every single second. You're getting impulses and you're deciding what to do. So these are very quick, brief reactions. Now, we don't want to keep sending indefinite signals, so that neurotransmitter is going to be quickly removed from the synapse either by reuptake or enzymatic activity. What I mean by reuptake is it is just taken in by the axon that releases it. So it's reabsorbed into the axon terminal that released it, or by enzymatic activity, or in other words, an enzyme breaks it down. A good example of this is when we went through our muscular system, we learned about a neurotransmitter called acetylcholine. And it was broken down by the enzyme acetylcholinesterase. Remember, if it ends in an ACE, it's typically going to be an enzyme. So whenever that neurotransmitter is broken down and detached, this causes the sodium channels to close so that sodium can no longer rush into the cell. And then from there, the system resets itself. So here's a little video of how synapses work that's provided by your textbook, just to kind of give you an idea.
Our brains are constantly processing information, which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron.
Okay, and that's kind of the end of what I was going to show you. If you have any questions, please reach out and let me know. And I hope you guys have a good day.